Hello. So, sir, should we start? It's the right time. A warm welcome to the third day of this 6-day FDP on revolutionizing healthcare. Today, as our first speaker, we have Dr. Rituraj, who is a senior principal scientist in the Department of Biotechnology at CSIR Institute of Himalayan Bioresource Technology, Palampur, Himachal Pradesh. He holds a B Pharma from B.R. Nahata College of Pharmacy, an M.Tech in Bioinformatics from the Indian Institute of Information Technology, Allahabad, and a PhD in Bioinformatics from Vellore Institute of Technology. He holds a postdoctoral experience from Italian Institute for Genomic Medicine, Torino, Italy, and he has previously served as a faculty member at Vellore Institute of Technology. Dr. Purohit has over 20 years of academic and research experience, which integrates computational biology, structural bioinformatics, and molecular simulation to address challenges in protein engineering, drug discovery, and therapeutic development. Dr. Purohit has authored over 150 plus peer-reviewed publications in reputed international journals and guided multiple PhD students. His research has significantly advanced understanding of molecular mechanisms in cancer, neurodegenerative diseases, viral infections, enzyme catalysis, with applications in healthcare, biotechnology, and bioresources. He continues to lead multidisciplinary projects aimed at rational drug design, biocatalyst engineering, and the valorization of Himalayan bioresources. So, we are fortunate enough to have him as a speaker today. So, now it is over to you, Dr. Purohit.
Am I audible?
Yes, yes, sir.
Okay. So I'm sharing my screen. So I hope my screen is visible. My screen is visible.
Yeah, yes, sir. Your screen is visible.
Yes, sir, it is
Okay,
visible.
fine. So good evening, everybody. So today we are going to learn a concept of very important methodology, which very frequently we use in drug discovery, drug design, or lead identification. That is called the QSAR, quantitative structure activity relationship. The basic concept of QSAR, generally, if you ask me in a very simple language, it is like that if you have the two molecule and they are very similar, say 95% or 98% similar, then we can easily say that both the molecule will be having similar kind of biological activity, right? So if the molecule looks like same, I mean they are very much similar in their structure, uh, in general the biological activity will be same, right? So that is the basic concept of QSAR. Whatever we have the molecule in our uh, I hope you understand what is the meaning of molecule and what is the meaning of if I am talking about biological activity, right? So the QSAR is completely the computational approach where we uh, generally observe the ligand database or ligand molecules, and we try to predict the biological activity without going for experimental validation, without going for an experimental observation like in vitro or in vivo experiments. So what is QSAR? So as I said before, that the QSAR generally we deal with the lot of chemical entities, chemical molecules. That chemical molecules or compounds or molecules, they are nothing but a ligand molecules which will be having some kind of biological activity while interacting with respective receptor, right? So is a computational modeling approach that establishes a quantitative correlation between chemical structure of a compound and their biological activity or physicochemical property using mathematical and statistical methods. I mean, so based on this methods, based on this concept that what kind of molecule we have, we will derive some kind of statistical parameters, right? And based on that statistical parameter, we will we calculate those parameters, and we after that we predict the some kind of biological activity. So in similar term, QSAR relates molecular descriptors such as hydrophobic, as I said, the different parameters, or you can say the proper parameters of evaluating a chemical molecule. That parameters is called hydrophobicity, electronic configuration of that molecule, static features. To the and that parameters relate with the biological activity of a molecule. QSAR attempts to find consistent relationship between biological activity and the physiochemical or structural property of a molecule, such as hydrophobicity, electronic effect, static factors, and topology. So, as in the picture, you can say that it's a very basic picture where a molecule having hydroxy group, right? And how it look like with that it will be having hydrophobic effect, right? What kind of static clashes if another group is there in this scaffold, there will be static clashes or not. That will everything we will evaluate in the QSAR, and after that we'll try to predict the biological activity of that particular compound. So this is kind of overview of QSAR, where we have the structure, experimental activities we'll be having, and similar for particular compound, we will be having descriptors of those particular compound, and based on that, we will have some kind of collection of those descriptors, and based on that, we will try to predict the activity of that structure, particular structure. The different we have different models in QSAR, and there are so many models, but very well-known models which we you can easily find out in any kind of QSAR book. The first model in that Hansch analysis, uh, where we relate biological activity to electronic, hydrophobic, and steric parameters. So this is one of the very oldest and most widely used QSAR method. It is two-dimensional QSAR method where we bother about the electronic configuration, hydrophobicity of the molecule, and the steric parameter of the molecule. Second one is the Free-Wilson model, uh, where express activity as a sum of fragment contributions. I mean, in that we will be having a fragment of the we will do the fragment of a particular molecule. For that, we would like to evaluate. I mean, the target we will we want to evaluate the biological activity. We will have some fragments, and each fragment, whatever the fragment contributes, that will be the biological activity of the molecules. So some fragments definitely will contribute. I mean, some fragment like contribute for hydrophobicity. Some fragment will contribute for their binding with the active residue, or you can say the particular residue. So that thing will have in this model, I mean, free Wilson model. Second one is third one is the topological QSAR, where we will be bother about the topology of the molecule, right? We'll have the graph, and we'll create the indices of that molecule, the connection between the each atom present in the molecule, and based on the molecular connectivity and the shape. So in that, we'll have the entire graph of the that molecule, and that graph contains information about the connectivity and the shape of that molecule. And the fourth one is QSPR. The P stands for the property here, where predicts the physicochemical properties instead of biological activity. So these are the four basic model. Through that, we generally observe the QSAR. We generally model our molecule for prediction biological activity. The first model, which we see in the previous slide, that is called the Hansch approach, attempts to relate the biological activity of compound to their physiochemical property using mathematical equation. It is one of the earliest and the most widely used 2D QSAR. As I said, in this, we will bother about the electronic, hydrophobic, and steric descriptors, and summation of all this will predict the biological activity of that particular molecule. But we have some limitations, like only consider first dimension or first dimension and second dimension molecular descriptor. We do not have any 3D descriptor information in this. Works based for conjugate series of compound, where the compound where we the conjugate series means the compound which are very much similar. If we have the diverse set of compound, it won't work, and cannot capture the complex interactions like uh, hydrogen bonding networks or receptor flexibility. That we cannot do with the help of this model, this approach. For that, we have the further approaches which can consider this type of bonding and the receptor flexibility. The second model which we saw, that is called the free Wilson approach, Free and Wilson, given in 1964, and it is a substituent based 2D QSAR method that quantifies the contribution of individual substituent at specific positions on a molecule to its biological activity. So as this approach basically assume that each substituent on the molecules contributes some kind of biological activity, and if like in this picture, you can say there will be if you want to predict the activity equals to some kind of constant, and contribution of substituent 1 R1, and the same contribution of substituent R2. So whatever the substituent there on the scaffold, we will consider each of one, and we will sum it. That will be the overall biological activity of that molecule. So it assumes that the overall activity is a linear sum of the effect of the substituents. So that it is not always that the all the substituent there on the scaffold will have positive effect. Some kind that definitely there will be some negative effect, like steric clashes between the two groups. And if that steric clashes is there, that will be also will be considered in that model. So we have the features. You can see the features of this approach is focus on the effect of individual substituents on the common molecule scaffold. Additive model, whatever the substituents are there, we'll edit it. Second one is the second QSR method. 2D QSR method. Again, use the structure descriptor like presence or absence of substituent rather than 3D. Again, we do not have any 3D descriptor or 3D three dimension component in this approach. Simple and intuitive, easy to understand and implement, especially for the small molecules. If again it is simple and intuitive, but again if it is the molecule is very big, this approach won't work, because we could not uh, uh, linear all the substituent effect in a through this approach. So in that way, for bigger molecule, this approach would not work, right? So a predictive tool can predict the activity of new analogs by altering the substituents. We have some limitations like previous one, they like ignores the interactions. We do not have the interaction between like synergistic or antagonistic or agonist effect due to the substituent. Assume linearity, assume effect of substituent are strictly additive, which may not always be true, as I said, for the bigger molecule. Not suitable for complex molecule, poor performance for molecules with multiple scaffold or complex conformation. So that time also this approach, the additive approach won't work, because the complex molecules will have multiple scaffold or two like antibody, or you can say the natural product molecule, where the molecules itself have very big structure, and each of the you can say substituent or the groups attached to the molecule having defined or definite kind of biological activity. So at that time also this approach will fail to evaluate the biological activity of that particular molecule. So we have so many, basically we have 6 type of QSAR. In that you can see that the based on the type of descriptors you are considering, like 1D, 2D, 3D, 4D, 5D, and 6D, 6 dimension QSAR we have. So in that, each of the QSAR is advanced, and currently we have 6 dimension QSAR, where we are considering the solvation model in the QSAR, and each of the QSAR is somewhat advanced. You can see the 5D QSAR is explicitly representing different induced fit model of 4D QSAR. So it is what, as it is said, that it represents the different induced fit model, means what kind of effect uh, occurs when a ligand or a substrate molecules um, uh, interact with the receptor. After the interaction, what kind of effect, that effect will be considered in the 5D QSAR, and that is called the induced suite mechanism. Before that, 4D QSAR, additionally including ensemble of ligand configuration. We in that QSAR we are including the ensembles of ligand, like different conformations of ligands, which is there in the 3 dimension. Like ligand can be active in one particular conformation, and that conformation can be captured at particular you can say the particular snapshot of ligand and protein. So that kind of information or descriptors will be considered in the 4D. 3D QSAR correlating activity with non-covalent interaction field surrounding the molecule. Like sometimes interaction field is very important, which you will see in the further slide. That kind of descriptors is there in the 3D, and the 1D, as we saw before, the few slides before, you have seen that about the 1 dimension descriptor, like pK or log p value of the hydrophobicity of that molecules, that will be there in the 1D QSAR. So these are the different types of QSAR. In that, each QSAR having some kind of advance, and the 6 dimension QSAR is one of the most advanced type of QSAR to evaluate the biological activity. So 2 dimension QSAR says that, like is a computation method that relate the biological activity of compound to their chemical structure using descriptor derived from 2D. molecular representation like topology of or physicochemical property without considering the three dimension. As we always consider, like whatever you see, like in this structure you can see the one benzene ring is there where you have two groups R1 and R2, and we will be having a value of electronic configuration of this molecule, hydrophobicity of that molecule, and steric, whatever the steric, if the steric clashes is there, then that will be also there in there. So this 2D informations will be considered in the two dimension QSAR. But in the 3D, we'll be having a different method, more specific method. You can say the 2D is 1D and 2D is I mean, there are the two major class, 1D and 2D, and the second one is 3D. So like 4D, fifth D, and 6D all comes under 3D. So in 3D, as I said, we'll be having three-dimensional information in this. It's an advanced computational method in drug design that analyzes how the three-dimensional structure properties of molecule influence their biological activity. And like 2D QSAR, which uses simple molecular description like hydrophobicity, electronics, and steric factors, 3D QSAR consider the spatial arrangement of atoms and their interaction with the target receptor. As you observed, or you, if you have done the docking or some kind of simulation, in that, uh, if you have if you see the trajectory of a protein ligand complex, you can see the if you visualize it in the video, then you can easily understand that ligand molecules continuously evolved with some kind of poses, right? And after some time, when the trajectory converged, you can easily see that ligand molecule stopped moving faster compared to previous one, right? So that configuration, or you can say that state is the most stable state, or you can say that state is the real state of interaction. And that information comes after, I mean, we have considered all kind of spatial arrangement of the ligand molecule, like what kind of atoms uh, present in the substituent R1, R2, or whatever they are in the group attached to the scaffold, and how that atoms interacting with the receptor amino acids, what kind of electronic contribution or electronic exchange occurring between the atoms involved in the ligand and the receptors. So that kind of informations we'll consider in the 3D QSAR. So in the 3D QSAR, again, they divided into three groups, like based on the intramolecular modeling, based on the chemometric methods, and based on the alignment criteria. So we will see in the detail in further slides, like ligand based, like COMSA, COMA, COMSIA, and JUM. Receptor based are the CORIA, combined, and FOMC. And apart from that, chemometric methods, linear 3D QSAR, and non-linear. And same way, the based on the alignment criteria, alignment dependent on COMSA, and COMSIA. That we will see the details of each of I think 1, 2, 3, and 4. 4 of this terminology modeling techniques we will see in the detail in further slides. This is the one of the analysis, QSAR analysis, where the comparative molecular field analysis will perform. As I said, that there will be some kind of electrostatic field around the ligand molecule, and that electrostatic field or charge field will consider to predict the biological activity. That comes under COMFA. It is called the comparative molecular field analysis. FA COMFA it means field analysis of that molecule. So how it works? The 3D QSAR method, COMFA correlate the three-dimension steric and electrostatic field of molecules with their biological activity. Molecular alignment requires all compounds to be aligned on a common structure framework before analysis. Calculates steric and electrostatic interactions at grid points around the molecule using a probe atom. There will be one kind of special atom, or you can say the common atom between all the ligands which we are aligning together, that atom, and around that atom will calculate the static and electrostatic interactions, and we'll generate 3D contour maps that you can see in the diagram, that show the region where the bulky or charge groups increase or decrease with the decrease activity. So in the visualization, or in this picture, you can see the contour map, where you can easily find which one is electronegativity, what is the where the electronegativity are increasing, where the other groups, or you can say positive groups, positive atoms are there. So that contour map will be generated for each of the molecule, and that contour map will be considered for comparison with the database molecules, right? So in that COMFA, we'll always consider the field of, or you can say the surround of electronic environment of the molecule. Second one is called the COMSIA, uh, where SA symbolize surface analysis. We'll bother about COMSA is a non-grid. Previous the COMFA was a grid based 3D QSAR, but in COMSA is a non-grid 3D QSAR approach that makes use of molecular surface by defining those regions of compound which are required to be compared using the mean electrostatic potential. We'll not consider all the surface, complete surface, but we bother about the surface which is important for the biological activity. The basic concept is that every molecule has 3D surface that interact with the biological target. Like the assumption of COMSA is like that, uh, whatever the interaction between the ligand and the receptor, that will be defined by, or you can say the response. The main key for the interaction is the surface, or you can say the environment uh, that particular molecule having. Environment means what are the donor, or you can say the acceptor groups present on the surface, right? What kind of interaction that particular ligands uh, perform in the receptor, that everything will be there on the surface. You can say the aura in the common language, the aura of that ligand molecule or compound will tells the biological activity. If the surface is perfectly fine, like balanced, proper electronegativity and donor and acceptors groups are proper, at the same time it should complement to the receptor, then you can say there will be very good biological activity. So the COMSA says, the overall you can summarize in that way, that COMSA says there will be some kind of surface of each of the molecules, and that surface will consider in the COMSA. Like COMSA examines surface properties such as electrostatic potential, hydrophobicity, molecular field strength. These surface values are compared across a set of compounds aligned in 3D space. Statistical method like PLS are used to correlate these surface descriptors with biological activity. This surface will tells about the formation of bond between the receptor and tail, and receptor and ligand, and that will decide the biological activity of that compound. What is the main difference between COMSIA and CORIA? So COMSIA compared the molecular similarity indices analysis, was mainly developed to overcome the certain limitation of COMFA, right? And the CORIA uses the descriptors that describes the thermodynamic event involved in the ligand binding. So CORIA is somewhat more advanced. Why? Because it considers the thermodynamic parameters when the ligand binds to the receptor. Like induced sweep mechanism, there will be thermodynamic events after binding, during binding and the after binding also. So for that kind of events, we'll apply the CORIA to evaluate the QSAR or to get the biological activity. In COMSIA, molecular similarity indices calculate from the modified seal similarity fields are employed a descriptor to simultaneously consider steric, electrostatic, hydrophobic, and hydrogen bonding properties. Some of the examples are given here. But in the Korea, the methodology simply consisted of calculating the non-bonded interaction energy between the ligand and the individual active site, right? Residues of the receptor which are involved in the interaction with the ligand. This approach was further extended and modified to develop two new variants of chorea, the reverse chorea and the mixed chorea. They are way of calculation, based on the way of calculation, they have divided into two, reverse and the mixed chorea. Now comes to the 4D QSAR. What are the fourth dimension which makes it 4D? Right. So 4D QSAR is the advanced QSAR technique that incorporates three spatial dimension like X, Y, Z of the molecular conformation, plus the fourth dimension representing the time dependent conformation flexibility and orientation sampling of the molecule. This is one of the very important criteria while doing the dynamics, right? You have seen that we will have the 3D configuration of each a trajectory with the time. So in 4D QSAR, we'll consider that constraint also, that after each of the steps like time, what will happen? What will happen to the molecule? How the three-dimension structure or 3D coordinates change? That kind of information will be considered in 4D QSAR. One factor driving the development of 4D QSAR analysis is the need to take into account multiple conformations first. Second one is alignments and substructure group in constructing QSAR model. As it is said that multiple conformation you need to capture while like if you want to have time dependent conformation flexibility analysis, you need to have multiple conformations, right? After that, you need to align those conformation if they are aligning, or you can say the convergence. Alignment is nothing but the convergence of the snapshots during the time, and the substructure groups in constructing, and the substructure groups, what are the structures, or you can say the part of the ligand having more movement, or you can say the part of the what are the portion where the ligand have rigidity. So that everything will be considered in this model. So this QSAR degree of freedom are normally all fixed in other 3D QSAR analysis. So far as 2D QSAR analysis can meaningfully predict the active conformation and the preferred alignment of a training set. So that for that we need to have dynamic constant in our process to get this the fourth dimension of fourth dimension descriptor of that to evaluate the 4D QSAR parameters. 5D. So another dimension is adding into the QSAR model, that is called the 5th D, and that incorporates induced sweet effect of ligand within the dynamic receptor environment. So we will be having the time dependent conformation. At the same time, we'll observe the induced sweet effect of the receptor, right? After coming ligand or substrate molecule, what kind of changes occurred in the receptor. So that kind of information will be considered as a descriptor in the 5D QSAR. So 5D QSAR accounts for multiple receptor conformations, typically from protein flexibility or induced sweet docking, effectively adding a fifth dimension, receptor adaptability. So that is the fifth dimension called the receptor adaptability. What kind of adaptive behavior occurs, or you can say the what are the adaptive conformations occurred after ligand interact with the receptor or substrate interact with the receptor. That kind of information will be considered in the fifth or a 5D QSAR. So how it works, workflow of 5D QSAR is like ligand and receptor preparation, conformation and orientation sampling, interaction field calculation, averaging across ensembles, statistical model development, and validation. These are the basic steps to evaluate the, you can say the fifth D, or you can say the 5D QSAR. In the, you can say you can see in the fourth step, averaging across ensembles, where this 5D dimensions, or you can say the descriptors coming, where we are taking the average, the field across the multiple ligand conformation, orientation, and receptor states. Another, or the, you can say the one of the latest QSAR, that is called the 6D QSAR, is the advanced QSAR methodology that extends the 5D QSAR by incorporating solvent effects. So this is another type of very important concept, is the solvent effect. All kind of reactions, or you can say the interaction, ligand protein or protein protein or protein RNA, protein DNA, macromolecular interactions occurs in some kind of environment, some kind of solvent. In general, in real time, it will be there will be cytosol, or you can you will be having some kind of environment solvent surrounding the surrounding this receptor or protein. So that effect, if you want to incorporate, that will be the sixth dimension, or you can say the six descriptors which we are incorporating in the to evaluating the QSAR. So, beyond the ligand flexibility, so ligand flexibility, other thing, beyond the ligand flexibility and time dependent dynamics and receptor flexibility, the 6D QSAR accounts for explicit and implicit solvent interaction, ion effect, and other environment influence that modulate uh, ligand receptor binding and activity. So if you have done molecular dynamic simulations in your PG or UG or some kind of project, in that you will be observing the two kind of environment, or you can say solvent, explicit and implicit. So in that way, you can incorporate the solvent into the system. There will be some kind of solvent which is surrounding to your complex. You are adding ions also in that to make it neutralize, or make it the pH which is very similar to the real, and other environment like pressure and temperature also. That everything we'll incorporate and try to capture the solvent effect to the ligand receptor complex, and that kind of information will be added into the QSAR model, and that will give the three-dimension QSAR. Ligand and receptor, so it will be more precise. The biological activity which you are having, which you will get biological activity, it will be more accurate, right? So in most of the drug discovery companies perform this kind of you can say the descriptor, to capture this type of descriptor. They used to perform dynamics of this complex in different way. We have the different approaches of molecular dynamic simulation, like metadynamics approach, absolute binding free energy approach, MMPBSA. So all these approaches are the are to capture the activity, or you can say the binding energy of between ligand and receptor, and that binding energy, you can say it could be directly related with the biological activity, because we assume that better the binding between the two entity like ligand receptor, better will be the uh, your biological activity. So this criteria is very important, and nowadays it will be very frequently using in very frequently in use in all by all the pharmaceutical companies, and you can say different companies, those who are predicting the new molecule, new leads by computational techniques, right? Solvent and environment incorporation include explicit water molecules, ions, other relevant environment factors during MD simulations. Sampling and interaction field calculation, compute 3D interactions field for all ligand receptor environment configurations, averaging across ensemble. So these are the way of doing or way of capturing the 6 dimension the sixth you can say dimension descriptor, a kind of descriptor solvation effect into the into for calculating this QSAR model. Method for building QSAR model, we have so many methods. Like these are the few methods which enlisted here. I'm not going to detail in the each of the methods. I'm just listed here, like linear regression analysis, simple linear regression, principal component analysis, partial least square analysis, genetic function approximation. These are the methods for building QSAR models. Application. Now, this is another interesting, you can say the why we are using QSAR, right? So QSAR, you know, I mean, till time, I mean, this time you understand what is QSAR, how we will do that, and what are the basic models, how it works, right? And now we'll see what are the application where we can apply this concept to have more precise result. As I said, the drug discovery and lead identification, predict biological activity of new compound based on the structure without going to the wet lab, right? This is one of the more interesting nowadays because we have the computational resources, and 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 we can easily avoid the expenses of performing experimental protocols or animal without killing the animal. We can have some kind of data, like we based on that data you can easily say that these are the potential leads that can be checked in the experimental platform, right? So nowadays you can say all the committees, those who are screening the projects, rather they recommend rather going directly to the animal or experimental. First you need to perform uh, computational platform. You need to perform some kind of computational experiments. Through that you can filter out the molecules which are not relevant to for your receptor or for the biological activity which desired biological activity. So so at the last, after performing this computational task, you will be having some kind of potential candidate that can be tested, right? So in that way you can save the money, save the time, as well as you can without sacrificing the animals, you can perform your experiment, or you can complete your experiment, right? So after that, I mean, the another application is toxicity prediction, estimate potential toxic effect of chemical through computational, reduces need of extensive animal testing, as I said, useful for environmental, chemical, industrial compounds, and the pesticide, right? So all these techniques, I mean, the QSAR, if you apply in toxicity prediction, again, you can save a lot of money as well as time and effort. So how it works, again, it will be having some kind of requirement, like if you are talking about the pesticide, or if you are working on some kind of identification of drug or lead, then there will be some criteria. This criteria should be there in your lead molecule. It will be there in the literature. So that information can be kept as a reference, and other thing you can based on that information you can process your data set. For data set, from that you want to pick the most potential pesticide or the drug molecule. So toxicity prediction also you can perform with the help of QSAR. ADMET prediction, this is another very lengthy, and you can say that tiresome job. If you are performing in the wet lab, you can perform in the dry lab with the help of QSAR, like evaluating absorption, distribution, metabolism, excitation, toxicity, because we have the very good models for each of the component. Like absorption, we have the very good model because we know anatomy of entire human body, how it will be absorbed, how particular molecule will absorb. If the hydrophobicity is hydrophobic groups are more, definitely it will be soluble. If lipophilicity, it will be soluble to the lipid. So everything is known. So if all the models are on are placed very well. So if you apply those prediction tools, and if you screen out the garbage from your database, then definitely you will be saving lot of time. Dictate poor pharmacokinetic behavior, because we need very good pharmacokinetic leads, right? So based on that, you can easily remove the molecules which are not performing good or having more toxicity, because we know that we have the models. Through that, we can easily predict the toxicity based on the, like, one of the very you can say the frequently used technique, or you can say the rule, is called the Lipinski rule. So based on that five criteria, you can screen your molecules, and that five criteria is how they are defining that criteria based on the literature. If the molecular weight is more than that, then it will be toxic. If it is not, so donor and acceptor is there, it could not be a lead. So exception is there, but mainly, I mean, if you see the major part, or 80%, you can say that 80% drugs follows the Lipinski rule. Maybe 20% or 15% won't follow, having the exception. But you can have that type of rules, or you can say the criteria to screen out your molecules from the database. The fourth one is the virtual screening and library prioritization. Again, you will be having big data sets. Like if you consider the pubchems, we will be having millions of molecules in that. And if you want to screen all of them for your particular activity, then definitely the QSAR model will help you to screen out those molecules. Regulatory and environmental application predict the hazards for chemical registration, assist in risk assessment and safety profiling. So everything you can perform with the help of this techniques, QSAR. Thank you very much. If you have any questions, we can have some more interaction. You can ask related to drug designing, or you can what I mean other things like how it works, how we can capture the descriptors from the MD simulations. What are the different techniques through that we can have, you can say, specific informations, like for evaluating the binding energy and other things. If you have any questions, you can ask. So it will be, I mean, interactive. I mean, uh, rather having more on that, I just given the basic overall uh, the overall view about the QSAR, and now it's up to you how, I mean, whatever you understand, you can ask. If you have some queries, then it will be more interactive rather than just one way lecture, right? I encourage everybody to ask questions.
Participants, if you have any queries, you can directly ask or write in chat box.
Yeah, you can write in chat box also.
There's a
Okay.
question that what parameters from MD simulation are required for 6D descriptor?
Okay, so 6D description, as we have discussed in this, like 6D descriptor is the solvation effect. So you can easily capture how your molecule or your complex, right, your protein ligand complex, or a molecule X perform in your particular environment, or with your solvent. So that kind of information you can capture while performing the MD simulation. with the water. Generally we will perform with the water. You can change other solvent also, like DMSO. Generally experimental we will do with the DMSO. You can have the DMSO also while doing the solubility analysis, and that effect you can easily capture after having the convergence of RMSD, and that snapshots you can take to have the effect of that particular solvent, right? Okay. One question is, can we use Korea and COMFA for peptide and ligand? For that, what particular changes we have to do? Okay, so for COMFA, it can be used for anything. I mean, any two molecules, whatever interact, like protein ligand, protein peptide, protein DNA, protein RNA, protein, everything you can have this mod. You can apply this model to capture the particular descriptor information. So changes based on your requirement, like if you performing protein and peptides, so that what kind of solvent suits for your complex, right? What kind of pH is required for that for your complex, as well as if you are having the if you are dealing with COMFA, then definitely you need to observe the you can say surface, or you can say the how the field of that your peptide. And the second one, what is the environment of your receptor cavity where the peptide or ligand interact. So that kind of information is required while dealing with the peptide and ligand.
Good evening, sir.
Yeah.
Sir, generally we do MD simulation of single protein and single ligand.
Mm-hmm.
Sir, if we want to do MD simulation of multiple ligand,
Okay.
single protein.
Multiple ligand at the same time.
Yes, sir, at the same time.
Multiple ligand, because ligands which you are doing, I mean, generally workflow says, I mean, first you need to perform docking, right? And docking predicts some kind of binding pose, right? You'll be having binding energy or binding affinity through that docking pose. You can do the ranking, right? Based on that ranking, whatever top 10 or top 20 complex, you can go for the MD, because MD is kind of extensive, or you can say laborious, computational laborious task. So if you perform multiple ligand in a one shot, it is not possible. Better you can have a one complex at a time, and you can have the you can observe the effect. Apart from that, what you can do, that if you want to do multiple ligand through MD, I mean effect of MD, better you capture the ensembles of pure, I mean only protein dynamics. Only protein dynamics, I mean you capture the ensemble of proteins while running single protein, right? Only receptor. And that best configuration of you can say the converged RMSD, you can pick say near about 100 conformations from that trajectory, and that 100 conformation you can do virtual screening for each of the ensemble, and you can have the best pose. So in that way you can do multiple ligand docking, multiple you can say multiple ligand simulation effect. That in that way you can capture.
Okay, sir. Okay, thank you so much, sir.
Okay.
So there is a question, can we use Korea COMFA for peptide
Yeah,
as well?
I answered that one. Any more questions? I encourage you all to ask questions so you can understand more things. You can ask any question not related to QSAR. You can ask any question related to drug discovery, drug
Sir,
binding.
good evening, sir.
Yeah, please, ma'am.
Sir, why it is necessarily to run the MD simulation program around 500 nanoseconds, sir? Why it
No,
cannot be done in 10 nanoseconds?
Fine. Very good question, and very informative for those who are doing this type of job. Why we required always higher end, why we want to do higher end? Like so many journals, they are asking perform 1000 nanosecond or 500 nanosecond. So the answer is that if you generally the physicochemical reactions, whatever reaction or you whatever the physicochemical effect you would like to observe through computational, generally it occurs in the real time with some time duration, like 1 second or 2 seconds. Sometime it will be more, right? To simulate that, we need to simulate our complex in till that duration. Like suppose if some reaction enzyme and substrate reaction is occurring for some some duration, like 1 minute, not 1 minute, like 1 second, or you can say the more than 1 second. So for that, to simulate, to capture the real time what is happening in reality, you need to perform the simulation for that duration, right? For 10 nanosecond is very small. 10 nanosecond in real time it won't occur. I mean that reaction won't occur. Some reactions definitely there will be there is a time that it can be done, but most of the things it will be go beyond the 10 nanosecond or 500 nanoseconds or something, sometime microsecond also. Another aspect is that while doing simulation, generally you observe the RMSD, right? If your system is converged, like if your RMSD is going after some time like this, and if you are going flat, there is no change in the RMSD value. At that point, if you are observing, if you are doing another analysis like RMSF or whatever the binding analysis or MMPBS, that will be relevant. In general, what happens for 10 nanosecond, generally system do not converge, and if upon that, if you are doing the prediction on that, it will be not good. I mean, it won't be correct. So best way is to perform the simulation till your RMSD converges perfectly, right? So nowadays the journals asking you need to perform triplicates. Like in experiment, we used to do triplicate analysis 3 times, like 4 times analysis, right? And after that you need to do average of all the triplicates. So that will be the best way to do MD, right?
Thank you, sir. Thank you so much.
Okay.
Excuse me, sir.
Yeah.
One more question.
Yeah,
Uh,
please.
what should be the average RMSD value that can be considered as a good range?
See, average RMSD, you can say the RMSD convergence criteria you are asking, right?
Yes, sir.
Yeah, so RMSD should not go beyond, you can say 1, 0.5 also. I mean, if you go, if it is going beyond 0.5,
In
then
nanometer.
yeah, nanometer. So if it is going beyond 5.5, then it is deviating. But if it is retaining zero point 0.5 RMSD till say near about 500 nanosecond, it will be maintaining between 0.6, 0.5, zero point 0.4. Then it is fine. It is converged. You can imagine it is converged, but it is keep on moving 0.5, then 0.6, then 0.7, 0.8. Then that RMSD is not good. You need to perform more simulation. You need to perform simulation for more time.
Okay. Thank you, sir.
Mhm. Any more questions?
There is a question.
Yeah,
Concept of molecular fingerprint, how it is used in QSAR and virtual screening.
yeah. Molecular fingerprint is nothing but you are taking the component of, like we have talked about. Like molecular fingerprint means what? That particular molecule having some kind of specific group that could be act as a that could be able to perform biological activity, right? So if we are taking that particular information, that is also very good. I mean, while considering the QSAR model, that information is also very good. And for virtual screening, I mean, that particular fingerprint, or you can say the key group, you can fix in the, you can say the, in the virtual scanning. While performing the virtual scanning, you can fix that particular group should not be changed. And because that particular group, or you can say the component, is the fingerprint of that molecule for biological activity. So while doing virtual screening, you need to screen your molecule, or you can say your database for that particular fingerprint. If and after that you collect all those molecules where you are having that fingerprint, and after that you can perform virtual screening in that. Any more questions?
Participants, do you have any more questions? Sir, I have a query. Like,
Yeah.
mostly when we do whatever we have done till now in QSAR, we have taken homologous series of compounds.
Mm-hmm.
Now, if we take structurally different compounds,
Mm-hmm.
then what would be the effect on the predicted activity?
It will deviate. I mean, like as I mentioned, that if we have the homologous conjugate series, then definitely you can perform the QSR with less effort. But when you have the different kind of molecules, then you need to have some kind of previous or literature information that what kind of molecules giving biological activity for that receptor which you are targeting. If you have that information, you can fix that information into the into while making the model, and and that model, or you can say that information can be run against the database of heterogenic compound. So based on that you can filter out.
Okay, thank you so much. Audience, do you have any questions? So if there is no question, then we can end this session and thank sir. So are there any questions?
Any more questions? You can ask a repeat question also. I mean, those who already asked, they can ask more questions, because it's not like that you are restricted like one question, two question. Dr. Uma, Dr. Srishti, Lovely, you can ask more questions. Shriyanki, Dr. Muhammad Atul Aslam, you can have more questions if you want.
So I think, sir,
Yeah.
we should end this session. Thank you
Mhm.
so much for joining with us today and giving
Mhm.
this information. No, we have a question. What is the
Mhm.
fate of classical QSAR in the era of machine learning?
Very good question. Uh, classical like QS concept of QSR is it won't be avoidable, right? We'll be having while doing the machine learning or AI, always we'll be having that fundamental model building way to perform QSAR, right? Machine learning and the AI and other things are the just techniques through that you can accelerate the way of doing, right? The method you can the method you can accelerate, but the classical or fundamental of QSAR will be the it won't be changed. The place won't be changed in the drug discovery technique. It will be there. It will be always there. It will be more faster. I mean, then, while applying the machine learning and the AI, it will be more faster.
So participants, any more questions?
Excuse me, sir.
Yeah.
Sir, is it necessary to do MMPBSA
Hmm. Yeah.
dissimulation?
Yeah. It's very necessary. I mean, very much required nowadays, because if you want to predict the binding free energy, MMPBS is one of the most frequently used technique. right? Without MMPBSA, if you want to predict the binding affinity based on the hydrogen bond analysis or RMSD, RMSF, PCA, or whatever the kind of analysis is there in the MD simulation, you cannot predict the exact binding free energy, right? The numbers. But if you apply this MMPBSA concept, it will be you will be having some kind of numerical value. Through that, you can say my ligand is ligand A is good compared to ligand B. Apart from MMPBSA, there are very powerful other techniques like ABFA, umbrella sampling is there. Nowadays, metadynamics is also used by the people, those who want to predict the binding free energy of the ligand and the receptor complex. So MMPBSA is required.
Okay. Sir, is it necessary to see that the same residue which was
Mhm.
we obtain in docking
Mhm.
should also give the good value or energy value in the MMPDSA?
No, it is not necessary because you are observing the dynamic system, right? But you should always keep in mind the residue which is responsible for biological activity, right? That should be there in the MMPBSA.
Okay.
If you are getting very good activity for
Okay.
a compound A, and it is not performing any kind of interaction with the key residue which is given in the literature, if there is no information in the literature, you can report that thing. But if there is the information in the literature, like particular FDA approved drug in the co-crystal structure having this residue interacting with the ligand and giving the biological some kind of biological activity, and if your ligand is making same bond with the with the receptor residue, then definitely it is important. If it is not, then you can say that activity could not be replicate, or you won't observe in the experiment,
Okay. Okay.
right?
Okay.
Mhm.
So there is a question in chat box by Lovely Run.
Uh-huh.
Challenges that arise using 3D descriptors in COMFA.
Challenges that arise using 3D descriptor and so challenges is to get the three dimension, I mean that descriptor information, like if you want to have comfa, then you need to have the you can say the surrounding fragment, right? The fragment, I mean the field analysis, field information of that molecule. If we have proper field information of that molecule, for that also we have different tools to get the field information. If you have the field inform, that is only challenge, to have the proper field, I mean correct field of that particular ligand or peptide or interacting macromolecule or a small molecule information. If you have the correct information, you will be having correct biological activity. That is only challenge, I mean, to get the proper field information of that ligand molecule. And another question is, which is the best virtual screening package for multiple ligand with single protein particular open source? Okay, so we have the Autodoc Vina, which is perfectly well on the place. I mean, it is performing very good for multiple ligand. Apart from that, you have Gunia, another tool is which is very recent. I think in published in 2024 or 25. That is called the Gunia, GUINNA. So you can type. It is also for multiple ligand, but well known is Autodoc Vina, and it is well established. It has very you can say the compatible with the Linux, Ubuntu, and other things. So you can easily install and perform your task. Okay, another question having how were protonation state of residue and ligand tautomerism considered for docking and MD? Okay, so for docking, protonation state is automatic while doing your docking protocol. Autodoc will be having one step like define the protonation state of that residue. Again, for this also you need to have literature information that if the residue like histidine or aspartic residue interacting while interaction between ligand and the receptor. So that protonation state you need to define consciously. Otherwise, automatically you can go for whatever they described by the software. So this is state this approach. I mean, for this step, you can have in this. It is there in the Discovery Studio or Schrodinger or Autodoc. There will be one step that will automatically assign the protonation state if you do not have any prior literature knowledge. Otherwise, you need to do manually. Any more questions?
So I think question answer session is about to be concluded. And I would like to thank you, Dr. Purohit, on behalf of the organizing committee. No, we have one more question. How do you reconcile the discrepancies between docking energies, molecular dynamic energies, and MMPBSA?
Okay, this is another very good question. That sometimes people confuse with docking energy, MD energy. MD energy is nothing but you can okay, MD energy and MMPBSA. So docking energy is the binding energy of that static pose, comes from the comes after docking. It is not dynamic pose, right? You'll be having some kind of interaction between ligand and the receptor, and that static pose having some kind of energy, that is static, it is not dynamic, right? While doing the MMPBSA, while doing the dynamics, you will be having energy of energy profile of that ligand with some kind of criteria. I mean, it is not the proper calculated, or you can say the based on some kind of principles. It has some energy based on how with the temperature how it is going, but again it is different from the MMPBS and the docking energy. The MD energy which you are talking, it is different from both the energy. While doing the MMPBSA is quite somewhat advanced from docking energy and MD energy, because it has some kind of fundamental principle. Through that, they evaluate this you can say solvation effect, dissolution effect, van der Waals effect. Everything will be considered during this calculation, and that's why it is somewhat advanced in this three energies, right? I hope you understand. Okay.
So, dear participants, do you have any more questions?
Okay, another question: should we choose our complex for MD simulation based on MMPBS or docking energy? Definitely, for MD simulation, you need to choose your complex based on the docking energy, right? Because MMPBSA can be performed after MD simulation, right? So first, you need to rank your ligand molecule based on the docking energy. After that, you perform MD simulation for top 10 or top 5 for 5 complex, and based on that, based on MMP perform MMPBSA for those complexes, and based on that MMPBSA you can rank for the. So you need to choose your complex or best lead based on the MMPBSA. Sometime what occurs, like you will be having ligand molecule having least docking energy, right? I mean, it is in the 5th rank or 10th rank, but while doing MD, the MMPBSA energy is coming very good, so that is more reliable. I mean, that at that point you need to consider, or you need to rank that molecule first, which is having more MMPBSA energy, right?
So, are there any more questions? So now we would like to end this session with a note of thanks to Dr. Purohit Dr. Rituraj Purohit, we are very much thankful to you for sparing your time today and discussing this very essential and informative topic of bioinformatics, or whatever we call it, computer-aided drug design. So I thank you on behalf of the organizers and for such an informative session, and thereafter such an interactive question and answer session.
Okay. Thank you, ma'am.
Thank you so much. So, we have with us our next speaker, Dr. Muhammad Ataul Islam, who is Director and Head of Science, Silico Scientia Private Limited, Bangalore. His expertise include computational chemistry, computational biology, bioinformatics, and computer-aided drug design. He is Head of Science in Silico Scientia since August 24. From April 21 to July 24, he was a senior computational chemist at 3Bigs Omics Score, Bangalore. From January 2019 to March 21, he was a Newton International Fellow at University of Manchester, United Kingdom. During his stay at South Africa, he was a postdoctoral researcher in University of Pretoria. He has been an assistant professor for about 1 year at Department of Bioinformatics, Bharati Vidyapeeth, Deemed University, Pune. He has been a visiting faculty of Birla Institute of Technology, Kolkata campus. His educational background explains that he has done his PhD in computational drug discovery from Department of Chemical Technology, University of Calcutta. He has done his master's in bioinformatics from West Bengal University of Technology, master's in bioinformatics from Sikkim Manipal University of Health. He was awarded Young Scientist Award by Indian Chemical Society in 2007. He received a postdoctoral fellowship from Government of South Africa, Vice Chancellor's postdoctoral fellowship from University of Pretoria, South Africa, and he has, to his credit, 65 peer-reviewed publications and has been giving invited talks in more than 30 conferences. So now we have with us Dr. Muhammad Ataul Islam.
Thank you so much, Dr. Nidhi ma'am, for the kind introduction. And I'd like to welcome all of you and good evening to all. So let me share my screen first. Is my screen visible?
Yes, your screen is visible. If you can make it, yeah.
Oh, thank you. So today, actually, I was attending the previous lecture, that is excellent lecture by Dr. Purohit, and it was nicely explained about the QSAR and all these things. So I think this the mature level, that is the extensive level of the QSAR kind of the machine learning. So that actually I'll talk today. So rather I am focusing today on how focusing on particular approach. I would like to explain how the artificial intelligence and machine learning change the discovery, how it accelerate the computational drug discovery approach to minimize the time, minimize the effort, etc. So we know very well that is the drug discovery is a very complicated process, and it starts from the disease identification. And once you have the disease identification, you have to understand the mechanism of action. how actually it is propagating. And then once you know the mechanism of action, then need to go for the identification of the target and followed by the validation. Once I have a target, that is what are the target responsible for this disease propagation, we need to go for the screening of the molecule for that particular site. And then further, we go for the optimization and the preclinical testing, and then go for the regulatory like preclinical study, clinical design, phase 1, phase 2, phase 3. All these actually the traditional way of the drug discovery approach. So yeah, we know that is very complicated method. So here, if you see, there is the how it is complicated and how much time is required. So on and around, if you have the good funding, good infrastructure. So from beginning to end, it takes around 15 years of time to find out one drug molecule. And here the success rate is extremely low that I talk. So overall, drug discovery is not an easy task. It is a very complicated and trial and error based method, and and the failure rate is extremely high. So I say the cost is extremely high, around 2 to 3 billion US dollar required per molecule. Time, yes, I mentioned around 15 years of time, and the success rate definitely low, less than 1%. You can say maybe less than that. And a lot of experimental validations are required into the animal studies, human trials, etc. So in this situation, the computational play a major role. So when in the traditional drug discovery, the different computational involved and different approaches are came, so drug discovery gained the momentum. So here, these are the different approaches for the target identification. You can go for target fishing, the reverse target identification based on the molecule, or you can have the protein database screening. For the lead identification, that means designing the molecule, a lot of things are there like structure-based, ligand-based, that and all of QSAR, including that pharmacophore modeling or molecular docking or de novo design, etc. And once you have your lead-like molecule or the promising molecule, then you need to go for the optimization. Here, another set of computational approach you can use, the virtual screening or the pharmacokinetics or the different calculations of the different properties or drug likeness analysis. So these things you can do, and based on that, you can optimize, you change to improve the molecule. And once you have the lead optimization done, you can go for the preclinical trials, and there you can analyze the data using the computational method. So this, why I'm explaining slide, because I would like to emphasize that is what are the current available computational drug discovery mostly used. Beyond this slide, a lot of approaches are there also. But here, so once it is got the involvement of the computational approach in the traditional drug discovery, like Turtle got the rocket to for the drug discovery approach. So it reduced the time, it is a lot of that is investment and all these things. So now, actually, I'm going to explain the basic concept of the artificial intelligence and the machine learning, and then how we can use these machine learning thing in the drug discovery approach. So here, artificial intelligence is the branch of computer science that studies the machine intelligence. So nowadays, every day from morning to evening, evening to morning, you are surrounded by AI. So all the time, because without AI, we can't move a step ahead. When we're watching some YouTube video, automatically after some time the similar kind of video are suggesting here. Suppose you're talking, we are talking some we are doing some discussion, and my phone is just beside me. If you after that, if you open the Facebook, you will suggest such kind of similar kind of advertisement or things related to the discussion. So always there we are surrounded by the artificial intelligence, by machines. So we are not, as if I say in a bad way, we are not safe. So whatever you're talking, whatever you're walking, whatever eating, everything is monitored by the AI right now. So similarly, the how AI changed the drug discovery field, that actually I would like to mention today. I like to explain rather than any particular approach. So AI enhances the human decision-making drug discovery, but does not replace the expertise of scientists or physicians, definitely. And the most effective approach combines the human creativity, and it reduces the cost, shorten the timeline, etc. Definitely. So, in a different way, artificial intelligence is a simulation of the human intelligence in mechanics to perform tasks such as the reasoning, problem solving, etc. And machine learning is a part of the artificial intelligence, which enables or which learn the knowledge or get the knowledge from the data. And then through that, it can use for the unknown data. So then the deep learning is a part of the machine learning, and it is regenerating, developing the model of the data in the multiple layer in the neural network. So if I say that this machine learning, deep learning, these are the play or game of data. So in this current scenario, if you have the data, that means you are rich in case of information, as well as in case of money also, because this era is only the era of data. Maybe the data from the biological science, maybe the chemical science, maybe the pharmaceutical science, maybe the any business, or maybe anywhere. If you have the data, and from that data, if you extract the information, the bad information, if you extract the good information over the bad information, then you are economically you are rich, and definitely the information is also you are rich. So this data is the era of the data. So here, machine learning, there's a lot of, there is mainly 3 subtypes are there, like supervised learning and unsupervised learning or reinforcement learning. And then there are subcategories are there, like dimension reduction, clustering, classification, regression. And then there are other subtypes are there. So these are the subtypes, depends upon which subtype or which approach we can use in our research or in our data. That depends upon the 2 things. One is what kind of data you have, and what is the objective of your research, what exactly you would like to expect from the data. Based on that, you can select the data. Sometimes you go for the clustering, sometimes you go for regression, maybe the dimension reduction, or the classification, etc., etc. So these are actually the subtypes or the domains or the fields of machine learning. So apart from that machine learning, the other approaches are also used in the drug discovery, like deep learning, I mentioned, natural language processing, the NLP, reinforcement learning, generative AI. So these are the different approaches of the artificial intelligence are widely used in the drug discovery. Again, I would like to repeat which approach I would like to go. That depends upon your data and your objective of the research. So definitely, when you have the target identification, a traditional approach, it is slow and manual experiments are required. But when you can implement the AI-based approach, so it is speed up the prediction. Similarly, virtual screening also, it is actually reduce the time, and you can explore the large chemical space to screen the molecule. Optimization stage also, it is faster. And in case of the preclinical, like pharmacokinetics and all these things, you can use and artificial intelligence. And overall, it is reduce the time and cost. So in a healthcare sector, along with the other sectors like business or management or any road safety, maybe any other fields, the AI is hugely using in the healthcare sector. So like clinical trial and monitoring, quality assurance, quality control, pharmaceutical manufacturing, pharmaceutical product development, and pharmaceutical product management, and obviously in the drug discovery. So today, actually, we try to understand how the AI or the machine learning transform the drug discovery in a new dimension. So here you can use the targeted protein prediction or the de novo design, bioactive prediction, toxicity prediction, or physicochemical property. So these things, mainly the extensive of the QSAR that is from the last lecture. So here AI can be used a repurposing approach, that is existing drug you can use. I will show the real success, one of the drug repurposing approach using the AI. Target identification, validation, designing the molecule screening, definitely optimization, biomarker discovery, clinical trials. So these are the fields where, so already the computational approaches are there, and now when AI came in the market, that it get the new what I can say momentum, or the exploration space increase use. And if you explore in a large quantity of data, if you can expand the chemical space in a wider space, definitely you will be the information rich, and that will help for the decision. So here, actually, I will explain few approaches. That is how artificial intelligence are using in the drug discovery. So a few moments back, I explained that the data is the main thing in the machine learning. So depending upon data, you can develop your model. Depending upon data, you can extract the features. Depending upon the data, you can extract the information, and that information you can implement in the unknown data. Okay, so similarly in the QSAR, when you have the known molecules or known data, from there you can correlate with the biological activity with the chemical descriptors. And once the model developed from the known data, and that model you can use to predict the biological activity of the unknown data, or you can design the new molecule from that model. So similarly here, I need the primary data. So primary data in the case of the drug discovery, definitely it is the molecule. So the molecule you can extract from the different databases, public laboratory databases like Campbell, PubChem, Drug Bank, Coconut database, SNS database, Zinc database, or may have your own data. Suppose you have a lot of data in your lab, that also you can use for this study. So once you have the data, then you can generate the descriptors. We already learned the previous lecture, that is from 0 to 6 descriptors we can implement. So once we have the data in the form of, that is the descriptors. Descriptors is nothing but the numerical representation of the properties of the molecule. So once you have the data, then you can use those data to develop the models. So from data, you will generate the descriptors, and these descriptors you can implement in the model. So depending upon your data, type of data, and exactly what exactly you would like to go, so you can choose the model, that is regression model or classification model or dimensionality model or clustering model or what. Once you have the model, and that model you can implement based on the different field like drug discovery or designing a new novel molecule, or using the clinical trial designing, toxicity prediction, or bioactivity prediction, etc. So this slide explain that is how you can develop the machine learning model and what kind of data you need. That data transfer into the data of properties or data of descriptors, and that descriptors you can use, that descriptor you can use for the model development. So here few applications, like in case of like binding affinity calculation. So here I'll explain shortly how artificial intelligence or machine learning are using in this machine learning. That is, suppose you have a huge number of molecules to dock or to calculate the binding affinity or binding energy. So suppose if you have the hundreds, thousands, 1 lakh, 2 lakh, that can be manageable. maybe 1 million manageable. Suppose you have 2-3 billions compound. So what to do? So what actually we do, we calculate, we dock the smaller set of molecules, and based on that binding affinity, we develop the model and predict the binding affinity of the remaining molecule. So that I'll explain shortly. Then again, drug target interactions. So we can understand the known data from the protein ligand binding interaction data. We learn it, we predict the model, and once you have the model, you can use that pattern, that model in the unknown complexes to get the detailed information of the protein and ligand interaction. Designing of the novel molecules similar like that, if you have known molecules, from there you extract the data, what kind of features or what kind of properties make them the potentiality for certain target. So those features will extract and develop the model, and from there we can design the new molecules. Okay, sorry. So then you can design the new molecule, and in here the target structure prediction. This is a very good example. That is, we need the 3D structure of your protein target for the in silico study. So if you have sequence, how can you develop the 3D structure? So homology modeling is one of the approach. Here also you can use the AI machine learning. So I'll tell you maybe the later stage. That is how AI are implemented to convert the sequence into 3D structure and get an excellent job by the Baker's lab and others and got the Nobel Prize. So this way you can use. So this is the only 4 approaches are there. The much bigger approach are there, like virtual scanning, all these things I'll talk about later this time. So here, in case of the pharmaceutical analysis, like toxicity prediction, if you have the known data, toxic data, non-toxic data, and then from that data you extract the features and develop the model, and that model can be used for the known molecule to segregate whether it is toxic or non-toxic in nature. So in bioactivity, we have known molecules, develop the model, and based on the model, you can identify the new molecule, maybe the anti-cancer, antibacterial, or any kind of that. Also, for calculus of the physicochemical properties, suppose you have a large number of molecules, you would like to check, you like to find out the important physicochemical properties. So that also you can develop from the known data and develop the model and calculate the physicochemical properties. So overall, this actually, so now I'll just one by one, I'll just explain that what are the things required for the machine learning for the drug discovery. So here the target identification and validation, like we need the integrated genomic or transcriptomic proteomics data, and it detects the pattern to reveal novel biological targets, and AI models identify key mutation and pathways linked to disease, helps prioritize the target most likely to respond to the therapy. So as I mentioned earlier, that is how machine learning can be used in the docking. So deep docking is one of the good tool. It came, I think, 2023, I think, if I'm not wrong. So what exactly here we do, people do, that I mentioned, I have 5 lakhs molecule. So for my normal system, it will take time to dock. So out of 5 lakhs molecules, suppose I consider 30,000 molecules. I select randomly 30,000 molecules. I dock these 30,000 molecules in a traditional way molecular docking I have done. Because compared to 5 lakhs, 30,000 is easier to dock. Once the docking done, then we can develop the model from the molecular features as well as the binding affinity. Once I have the validated model, that model I can use to predict the remaining 450,000 molecules binding affinity. So we no need to dock those 450,000 molecules. So this way we can use the machine learning approach in the docking or calculation of the binding affinity. Similarly, in binding interaction pattern also we can predict here. The screening and designing, definitely, if you have a large number of database, suppose you would like to screen the entire PubChem or entire zinc database. So how we can you do? You can develop some models, and then based on the models, you can screen your molecules. So already we have done some work in our current organization. Pharmacokinetics prediction, definitely it is a QSAR, the quantitative structure toxicity prediction tool that is developed through the machine learning to extract the features, use the different machine learning algorithms, and then we can predict the different pharmacokinetics parameters. The drug repurposing, this is the excellent approach. That is, how can you use the existing drug for any existing approved drug, approved drugs for the other disease, to treat the other disease, that the drug repurposing approach you can use. So I'll show you one of the real example here. So here also you can use the molecule for the new therapeutic for new disease. De novo design, it is a very good approach. So to design the novel molecule or the new molecule for any target, you can use a generative AI model to build the molecules, and that molecules can be screened for certain target, and you can get. So already we have done some couple of work. I will show you some maybe 2 case studies. So here target specific ligands you can use, or scaffold hopping also you can do, and then after that you can implement the docking, MD, and the other analysis to narrow down the molecules. Clinical trial study also, the different stages like regular submission, data analysis, data management, supply management. So here also the existing data you can extract, and based on that you can develop the model and predict how can you optimize the clinical trials as well. So big data that I told you, that the data is, if you have the data, you are the rich in both economically and information wise. So you can handle the data from metabolomics, genomics, medical imaging, proteomics, and then these data you can use in the AI integration, and then you can get the solution. The precision medicine also, that you can use the AI, like you can use the genomic data or the clinical data, and then adequate tumor mutations or the drug response, and then the genetic variation. All these things you can implement in the machine learning approach. So in our current organization, we have published around 20 articles in the last 2 years, the combining of the traditional drug discovery as well as the machine learning. So advantages, definitely faster and cheaper, higher in accuracy, big data you can handle in an easier way, because in a traditional method there is some limitation to handle the data that can be overcome by the computational approach. But also the computational approach also there is some limitation that limitation can be break by the involvement of the machine learning here. So novel molecule generation, target identification, drug repurposing, all these things you can do that actually already explained. Now I would like to explain that is how the machine learning came and what is the current scenario and how people are crazy about it in the industrial sector or the academic sector, and how people are investing. So that I'd like to explain. I would like to take you, that is how machine learning are more important in the current scenario in the drug discovery field. So in the 1950s, the concept of the classical QSAR is where you can say the origin or the fundamental thing of the machine learning. then once the memory is increasing or the compressor configuration is increasing, the different drug discovery approach are coming, like comparative drug designing, docking, variable selection, artificial neural network, then applicability of various other domains, deep learning, deep neural network. So these, depending upon on the how the computational power from increasing, like CPU to GPU to multi GPU to cloud. So this is data as old data around 2010, but the current scenario is much bigger. So how once the computational power is increasing, their size is increasing, the involvement of machine learning in the drug discovery is increasing and doing the tremendous job. So here, the involvement of artificial intelligence in drug discovery, the total market size, it is, I think, 2023 data. So 29.7% in global market, and you can see that 2023 it was at 1.5 billion US dollar. And what is the projection? A different field like oncology or neurodegenerative disease or metabolic disease or cardiovascular disease. So people understood the essence or the beauty or the real application of the artificial intelligence drug discovery. That's why it is, you can see the market size, how the market size increasing rapidly. So here in a process-wise, like drug optimization, repurposing, or picking testing, and others, these are the involvement of the total market size. So most of the people are investing in the drug optimization and repurposing field. So here you can see these circles actually. So in each circle, the list of companies or the research organizations or names are there. Those are currently involved in the artificial intelligence drug discovery. So if I zoom it in a 400 or 500%, even you can't read the names. So you can understand that is how much companies or how much the research institution, they are jumping in this field for the application of the artificial intelligence drug discovery. And this is the old data. So the big companies as well as the small companies, they are doing the excellent job in the field of the artificial intelligence drug discovery. So these are some milestones already that is it is proven that yes, the artificial intelligence can design or develop some molecules like, but it's that I'll explain also. There is a benevolent AI that is the purpose for COVID-19 and is FDA have given the approval, I think, in 2022. Like LYCOV1401, this is the Accelera. They use the AI-based approach and this is in the clinical trials. So here, like BXCL, this is also phase 1 or 2. This is when 2293. This is also relevant AI. It is phase 2. Like this, these are molecules. All molecules by the different companies are designed or identified or screened through the help of AI machine learning approach. So here, in a more like here, you can see that that INS 018055, this molecule designed by the in silico medicine, and they use their AI platform, their own AI platform, and that reduced the time at 4 to 5 years. Similarly, benevolent AI that I already mentioned here, that is the previous slide, the baricitinib, that was actually for the arthritis drug, but it was repurposed for the COVID-19 and even they got the approval from the FDA, and that also developed using the AI platform. Exscientia, they designed the drug candidates like DSP1181 by the artificial intelligence. Like predicting the different side effects that Atomwise is doing the excellent work. They are using their deep learning platform, the ADMET, to predict the binding affinities and the molecular targets, helping reduce the candidates with a high toxicity and poor efficacy. So these are the some real outcome or validity of the AI in the involvement in the drug discovery. So it is not like that. That is, yes, people are talking about much about artificial intelligence. There is no groundwork, but yes, these are the validation of the artificial intelligence, how involving in a real-time medicine development. So here in the history, the first time the people got the computational chemist or the, you can say, the bioinformatician, they got Nobel Prize for their 3D structure prediction. So like David Baker lab and the Demis and the John, they actually, this AlphaFold with a tremendous discovery. This is for the structural biology, all the sequences available for the protein structure or the any they converted to the 3D structure, and the excellent job, and that recognized, and the Nobel Committee, they recognized their work and they got a Nobel Prize. And this is history, that is computational chemist or computational biologist. They got a Nobel Prize first time in history in 2024. So similarly, that is in our company, we are doing such kind of the mix of the traditional drug discovery, traditional computational drug discovery, as well as the artificial intelligence. So one of the examples here, we used the ETHR inhibitors for the MTB. So here we use the generative AI approach to design some molecule. We are one of the tool called Reinvent that came, I think, 2023 or 2024. So we use that package and design the large number of molecules, and from there we use the different AI machine learning approach as well as the physics-based approach like VINA or plant, and then we finally got the MD simulation, and finally from there, these are the molecules we got as a novel molecule. We checked that yes, these are the maybe the novel candidate for this target. Yeah, that also validated using the molecular dynamic simulation. You can see the energy plot of each of the molecule, and yes, we got excellent results from the computational study. And further, we are planning for the experimental validation of these molecules, and it is published the beginning of this year only. So another work, the same approach we used, and this is polyketide synthesis 13. So our research group worked on that, and using this generative AI to design the molecule. So these are the known reported PTS13 inhibitors. We use those molecules and generate around 50 to 60,000 new components, and we use the different physics-based method like VINA or plants, and then finalize these 6 molecules. And we also did the comparative analysis in the molecular dynamics simulation and document all these things. And this is also published, I think, last year or this year, maybe last year, I think so. So yes, there is no doubt, and it is well validated that is yes, AI can be implemented in a large extent in drug discovery. And people, if you just open your eyes, you can see, you can listen that yes, people are talking about AI, but there's a lot of challenges out there. So the artificial intelligence, or especially the machine learning, in the case of the drug discovery, the data is the main thing. If your data is good, you can predict a good model, and definitely you can predict the good molecule. But if your data is not good, then maybe you may be biased, or maybe you have the faulty results, or we have the false positive results. So that actually first thing you have to check your quality of the data, and definitely the size of the data. So sometimes people actually ask a lot of questions. That is, what could be the minimum size of the data? There is no limit, there is no rules at all. But if you increase the data, if it is a sample size or if it is a data size, so definitely you're covering the more chemical space, and that will help you to build the good model or the good predictive model. And also that is a lot of AI model. We got the model, but the interpretability is sometimes difficult. So if it is unable to interpret the model, then how can you understand that is? How can you take the decision, or how can you predict the good data about that model? So there is another problem is there. And that definitely the ethical or regulatory hurdle is there. Need multidisciplinary collaboration is required, like chemist, because chemists, they understand the chemistry, biologists, they understand the biology. So for them, it is very difficult to understand the extent level of the machine learning. So we need the collaborative work, multi-domain people we need, and that is another challenge, because when you explain the chemistry or biology to any IT people, they won't understand, and vice versa. So these are some challenges are there, but definitely the selection of the data, interpretation of the data, these are the main challenges in the drug discovery approach. So this is all about, that is, I told you, that is how the traditional drug discovery got the momentum through the computational drug discovery approach. A lot of computational drug discovery came, and now the involvement of artificial intelligence or machine learning in the computational drug discovery, it gave the new dimension. And the real example, reality works, and it shows that yes, this field is not only the myth, or not only that is figurative, but it is yes, it's a reality. Yes, people are getting the data, getting the approval, getting the molecule. So keeping in this mind, the few of us, we have thought that is yes, we should do something in this field, and we actually formed a new organization, that is the Silico Private Limited. So it is the artificial intelligence company, and we are working on the computational biology, computational chemistry, as well as the artificial intelligence in healthcare. So I am here. I am one of the co-founder and directors of this company. Along with me, Dr. Sonali Thorat. She is also the co-founder and director, and she is basically from chemical and pharmaceutical science. So we came together, we discussed it, and we formed this company. So it is 2 years plus company, and it is situated in Bangalore. So we are giving the services like computational drug discovery services. We are giving the solution of the bioinformatics. Also, we are giving the solution of the artificial intelligence in the healthcare. Apart from that, we are also providing the short-time to long-term training program to faculties or the scholars or the students who wants to learn about the computational drug discovery with the AI, or the computational biology with the AI. And also we're supporting the academic research support, like for publication. Suppose you need the publication, or you have the data, you have the project that you would like to execute it, then we also support that also here. So this is our team. So along with me, we have the Cheminformatics team, we have the AI machine learning team, we have the IT team, that is development team, and yeah, they are developed on the good platform. Along with these all services, we have developed on platform, that is complete one-line cloud-based drug discovery platform, that is a Silicon Flow. Because two things are there here. One is that the drug discovery is not difficult once you have the expertise. But problem is that you can do the computational drug discovery certain extent with your normal computational infrastructure as well as your technical background. But if you want to go beyond that, then you need the different platform, high computer performance, that is the HPC you need. And also you should have the knowledge of the basic understanding of Python programming or any language programming, or maybe the Linux platform. Because suppose I can manage 1,000 molecules manually. 10,000 molecules I can manually if I give my time very dedicatedly. But suppose you have millions to billions of molecules, how can you handle? So that time you need some computational programming language to deal that. So when I was meeting the different people from the chemistry or the pharmaceutical science or biological science, they are talking about this. There is how can I use the large number of molecules? I would like to explore entire database like Zinc database or Asenex database or NCI database. So how can do that? So to overcome this thing, what we have developed, that is in the banner name of Silico Explore, we developed our online cloud computing system. So here complete drug discovery tools are there. So here what we have done, we have around 27 tools. So few tools are the third-party tools; those are open source, and few tools are our own tool. We have developed a lot of tools through the machine learning approach. So a lot of like virtual scanning. If you have the large number of compounds, how can you use the machine learning to scan? So if you can come with the data like your known data, unknown data, and you just develop your machine learning model, and that model you can use to scan the data. So that tool we have. Toxicity prediction we have. That is, if you want to predict the toxicity, that is our machine learning tool. So this is these are tools, are the third-party tools we have integrated here. You can do docking in a large extent. You have to nothing. You just open it in your browser. You just upload your molecules and submit it. All the calculation will be done in the cloud system. So here the beauty of the system is that most of the tool you can do the docking one protein, one molecule, or one protein, multiple molecule. But here you can use the one protein, one molecule, one protein, multiple molecule, multiple protein, one molecule, multiple protein, multiple molecule. So one to one, one to many, many to one, many to many. Suppose you have 10 protein structure, you would like to talk 1 lakh molecules. You just upload this 10 protein structures, 1 lakh molecule, and submit it automatically one by one. It will go on and it will complete the entire process of the docking. So these other tools are there, and also the analysis. You have to nothing to do. Just click something and give the parameter. It will already screen and give the data. Similarly, we have the physicochemical property calculation. We have the QSAR, machine learning QSAR model. You come with your molecule, you develop your own QSAR model, and that model you can use to predict the unknown data. You have the DFT calculation, you can use the DFT of the molecule. Like this, also the rescoring is one of the important tool, that is our own tool, machine learning based tool. That is, after docking, you have the poses. But how do you know that this pose or this binding energy is not by chance, that it is not the false positive? How would you know? Because generally what we do, we dock the molecules, we take the check the binding energy. Binding is good, I can consider. But there's no option to check, cross check whether it is false positive or like that. So we have our own machine learning tool. We developed the rescoring. So you can use that pose and it will tell you after regression analysis, yes, whatever the prediction done by the docking, yes, it is the correct or not. So we already benchmarked this tool, and we found a lot of molecules even in the order of vena or the plant, they are generating the false positive. So that is very much important. So like that, we also have the peptide designing a tool with machine learning. Also integrated the molecular dynamics simulation like molecular dynamics GROMACS. So generally, the people are scared about the molecular dynamics simulation, especially the GROMACS, because a lot of commands are required, a lot of errors are coming. So that also we removed here. You have to upload it, the small molecule and the ligand, it's protein and ligand, and it automatically all the things will be done and analysis will be done. So this is a complete drug discovery platform, and it is online. There are no required. So here I the entire cycle, that is the drug discovery, how can you use this tool. So it is gap between the experimental and the in silico. It is a time to discovery, because the beauty is that there's no limit of using the molecules. Suppose you can use the 1 million, 2 million, 3 million, whatever the molecules you would like to, you can submit at a time and you can get this tool. So this tool already in market, we launched it, and also already a few universities took the license, and they are successfully running those tools, and yeah, it is already launched. And now, actually, very briefly, I would like to say that we understood the importance of the artificial intelligence, the next level of the use of artificial intelligence. If today I am not aware of the artificial intelligence, I have to learn up maybe in 1 or 2 years later, because everything is coming in the artificial intelligence or machine learning. So, like if I remember in my old days, there are people. Actually, when the computer came in the market, in a different sector, the people, those people or the old employees, they're supposed to learn the computer to work, to save their job. So that situation is coming right now. If today people are not learning this machine learning, maybe after 1 or 2 years or 3 years, they have to learn machine learning to save their job, because in every sector, not in drug discovery, everywhere, the drug discovery machine learning is applying and going to change everywhere in all sectors. So keeping in this mind, along with the one of the reputed university, DY Patil, Pune, we collaborated it as a industry partner. We launched 1-year diploma course, and that already started this year, and still admission is going on. So it's a 1-year program. So in this course, from beginning to end, entire thing, that is the programming language, machine learning, computational drug discovery, how can you use the machine learning, the computational drug discovery, that all are actually covered. So that when someone will complete this course, he or she will be ready for any industry appointment. Because as I say, if not today, maybe in a coming years, everyone should have to learn machine learning to survive in this sector. So all of this, okay, so I'm concluding. Yes, you explained about how the machine learning can already transform the drug discovery, and what is the going in the future, how the investors or the companies or the research organizations are jumping in this field to get the benefit of this machine learning in the drug discovery field. So thank you so much for your patience, and if you have any question, I'm happy to answer.
Participants, if you have any questions, you can directly ask or write in the chat box. Dr. Islam is there to answer your queries.
Yes, please.
Any queries regarding the presentation?
Sure, Safi, I'll send you my.
It appears it's.
Yeah, definitely. I'll share my.
If you have any questions, this is the last and final call. Participants, if you have any questions, you can write in the chat box or ask him directly. So I think it's dinner time for them. So
I can understand. But if you have any query, any doubt, because today I have not explained any methodology. I try to emphasize there is in the next coming years what is going to be happen. It is already started, so we have to transform with the time. And yes, machine learning is going to capture. And yes, it is no doubt it is a very essential in all sectors, including the drug discovery.
So I would like to congratulate you, Dr. Islam, for you people have started this advanced diploma in AI and computational drug discovery along with the D.Y. Patil University, Pune. So this is a step towards academia industry collaboration.
Exactly.
Yes.
Yeah.
So I appreciate these efforts of your company. And you have taken a lead in all this. I congratulate you.
Thank you so much.
And thank you so much for being with us this time again.
Yeah.
And I hope our association continues.
Definitely. Yeah.
So thank you so much.
Okay, thank you.
On behalf of organizing committee, I thank you.
Okay, thank you so much, and good.
Thank you, participants, and good night.